Welcome to the THOR Table of Knowledge podcast, where we dive deep into the groundbreaking world of trauma care, hemostasis, and oxygenation research. A special thank you to our sponsor, Velico, for their support. Velico is revolutionizing trauma care with their innovative frontline ODP spray-dried plasma system, with the goal of empowering blood centers worldwide to produce dried plasma for transfusions.
So, really interested to talk to you today about dried plasma. I want to start off with some background information on dried plasma, when it got started, back almost 100 years ago, and why it's so important for hemostatic resuscitation.
Right. So that's actually a fascinating story. Basically, in the aftermath of World War I, people had figured out that blood products generally, but specifically whole blood, was really important to resuscitate patients with hemorrhagic shock. Problem was they recognized that getting whole blood far forward in the pre-hospital setting was going to be a challenge. And right around that time, the development of freeze-drying technology was coming into industrial use. And so people had the idea of freeze-drying plasma as something that could be feasible to provide a blood product for resuscitation as a bridge to whole blood, or perhaps even a substitute for whole blood. It didn't really work out that way. We ended up producing in the United States something like 10 million units of dried plasma during World War II, and used it extensively, but also discovered, with the help of our British colleagues, that it functioned best when used early as a bridge to whole blood, because ultimately people need everything that's in the whole blood when they have shock. So that was kind of the beginning of the dried plasma story.
Fast forward to current times, there are now many different ways of producing dried plasma. Can you talk us through the different ways of manufacturing and how that might affect efficacy and safety?
Sure. So in the old days, in World War II, what the U.S. government sort of drafted pharmaceutical companies to make huge volumes. As I said earlier, over 10 million units produced. But the way they did this was they collected like tankers full of plasma and mixed it all together. And there was one small problem with that, which is that people didn't understand the potential for hepatitis to be transmitted by a blood product transfusion. And so one donor with hepatitis could contaminate, a 1,000-liter batch. And that's exactly what happened. So people stopped making dried plasma in the United States really after the Korean War because of that contamination issue in the manufacturing. Now, interestingly, our colleagues around the world did continue to make dried plasma. For example, the French military adopted a single or a mini pool approach. So what they would do is they would collect mini pools of 10 donors, and they would get some group A, some group B, and some group AB, kind of neutralizing the antibodies and making a universal product. And they would quarantine the product and then retest the donors and make sure that the donors were healthy and didn't have hepatitis. And once the tests for hepatitis and HIV and other transfusion-transmitted infections became available, obviously they implemented those tests. In fact, later on, the French military adopted another technology of using amotosalen and ultraviolet light to neutralize pathogens found in the plasma as an extra measure of security. So the bottom line is that there are different ways of manufacturing the plasma. There's how you collect the donors and pool them, whether you can make individual units of plasma from individual donors that you test and quarantine. You can use pathogen reduction technologies. There's another approach that's been used where people make large pools, but use a different pathogen reduction approach using solvents and detergents to get rid of enveloped viruses and nanofiltration to get rid of other viruses and prions and so forth. And that's been shown to be safe as well. So bottom line is what's involved is different strategies for single donor, small pools, large pools with pathogen reduction. And then the drying process has changed as well. So it all got started with freeze drying. But more recently, spray drying has also been introduced. So spray drying is used across the pharmaceutical industry as a way of drawing all sorts of products. And basically, you squirt the product you want to draw, in this case plasma, through a nozzle, atomizing, making very small droplets, and then hitting that with a blast of hot air, and that basically removes the water. And so there are two different ways of doing this, and both of those methods have proven successful. So right now, there are a couple of, so there's the German Red Cross, which manufactures a freeze-dried single donor product. There's the French military, which, as I mentioned, uses this mini pool approach and pathogen reduction freeze-dried product. And then there are several other companies that use different...
So there's Octapharma products?
There's the Octapharma product, which is licensed in Europe, which uses large pools, solvent detergent treatment, and lyophilization, freeze-drying. There's Teleflex under development in the United States, which uses single donor, non-pathogen reduced, freeze-dried. And then there's Velico Medical, which is building equipment to allow donor centers to spray dry a plasma product. Really, they could be of their choice. They could either do a single donor, a mini pool, or potentially even a large pool, and incorporate pathogen reduction as they wanted, theoretically.
And there's also Norway, I believe, working towards developing their own homegrown dried plasma product, so to speak.
Yes, which I believe also is based on freeze drying. Other people have experimented with, I should mention the biopharma product in South Africa, which is also licensed, which is a pooled solvent detergent product, as essentially been under license from Octapharma, as a very similar product.
Right.
So there have been different approaches. A number of other companies have attempted to make either spray-dried or freeze-dried products or equipment to make those things. But the currently functioning efforts right now are the ones by the German Red Cross, the French military, Octapharma, and Biopharma in South Africa, Teleflex, and Velico.
Great. So how about data on efficacy and safety with all of these different dried products? Can you update us on the current literature on efficacy and safety?
So from a safety standpoint, there's a fairly reasonable body of data coming from the French military and the German Red Cross. There's a lot of historical use of those products that suggest safety, really no significant transfusion reaction frequency reported. And the products do have the ability to restore hemostasis by provision of all the factors that are necessary in hemostasis. That's been extensively characterized. So that's all good. Products such as the Octapharma product are based on extensive use of the underlying solvent detergent plasma product, Octaplas LG, which has been transfused to millions of patients. And there's less data on the specifically dried and then reconstituted Octaplas, but clearly the underlying product is very safe. The data on the spray-dried Velico product is forthcoming. That is in clinical trials right now. And the company has completed 2 out of 3 cohorts in their initial safety study and is completing the final cohort. Results should be out in the fall of 2024.
But for the products that are currently used in Europe and other parts of the world, you mentioned the German Red Cross, the French product, used predominantly in France, and now this Octapharma product. Are there any large randomized controlled trials with those products, or have they been licensed without large trials?
Right. So this is where things get a little bit more complicated, in part because of the long history of use of these products, which has sort of allowed them to be grandfathered. The short answer is no, there are no large clinical trials, randomized trials with these products. However, there are some small randomized trials in France with the French freeze-dried product, again showing safety, some evidence of efficacy, such as, for example, able to restore fibrinogen levels in patients transfused the product who had massive bleeding. Now, from a randomized trial standpoint, there's been a lot of interest in getting to randomized trial data. The U.S. military, because of its interest in producing or in having people produce for it, dried plasma once again, has sponsored a number of these trials. In this case, using not dried plasma, but testing the concept of pre-hospital early transfusion of plasma, which is what the dried plasma would be designed to facilitate. Two of those trials were the COMBAT and PAMPER studies. COMBAT was a single-center study done at the University of Colorado. PAMPER was a multi-center study, pre-hospital helicopter evacuation of patients study run by University of Pittsburgh. The major difference, other than multi-center versus single-center, is that in Colorado, it was one hospital receiving patients from ground ambulances with very short evacuation times in the ballpark of 20 minutes versus pre-hospital times in the ballpark of 90 minutes, I believe, in the PAMPER study. And the nice thing about these two studies is that the DoD planned a pre-study harmonization process where they were able to pool all the data. And the bottom line is that PAMPER showed a significant reduction by about a third of mortality in patients receiving 2 units of plasma as their initial resuscitation compared to standard of care. COMBAT didn't show any significant difference, but then again, the evacuation times are so short that many of the patients never actually received the full intervention, so it's hard to say. When you pool all the data, basically, if patients had a greater than 20-minute evacuation time, they seem to derive a benefit from early plasma transfusion. Now, the one larger study that's been done actually with dried plasma was the refill study in the UK, which was a multi-center study looking at the combination of red cell concentrates plus the German Red Cross freeze-dried product, Lyoplast, versus saline, actually. And there was no statistically significant mortality benefit from the blood-based resuscitation versus the saline resuscitation. However, there were a couple of issues in the way the study was designed and enrolled. I mean, in particular, the fact that patients didn't receive the study interventions until about 50 minutes after injury, which is quite a bit longer than in either COMBAT or PAMPER, and perhaps longer than you would expect them to be able to derive a benefit from. The other thing is both groups got a lot of saline up front, so there might have been some dilution, if you will, of the signal. Recently, there was a meta-analysis published looking at all three of those randomized trials, which, as a meta-analysis goes, is not ideal since there are only 3 studies with a limited number of subjects. But the bottom line is that the authors concluded that there's probably a mortality benefit in the ballpark of maybe 15%, not statistically significant in the meta-analysis, suggesting a need, obviously, for more data, but directionally speaking, suggesting certainly no evidence of harm and probably benefit. Look, I think what the data show us, honestly, is that this is a safe product. It is likely to reduce mortality if used as intended, which is early blood-based resuscitation to support hemostasis. And by early, within certainly that first hour, preferably as soon as possible following injury, as the military data have shown us, and that it will effectively serve as a bridge to whole blood. The military is very interested in this because one of the huge challenges NATO countries and others face is the problem of readiness. When forces are not in conflict, when they're sort of serving a deterrence mission, there's no trauma going on, there's no bleeding happening, and keeping huge stocks of labile blood products on the shelf would lead to shortages because it would all be wasted. Whereas the nice thing about dried plasma is that it allows you to keep a product that can serve as a bridge to full resuscitation for much longer periods of time in a convenient form with a much more convenient cold chain so that you have something on hand in case something happens. And that's also true for civilian EMS agencies when we think about disaster medicine or simply care of patients in rural areas where, you don't have a huge volume, but you may face long evacuation times and people need support during that evacuation. This is a product that could revolutionize the care of those patients.
Great. So you've focused primarily on pre-hospital utilization of dried plasma, which makes complete sense. I'm going to ask you a series of short questions that we're going to get short answers for, that leading to the main question of its utility in hospital. Okay, so the first question is, what are the reconstitution times for the different dried plasma products that you've mentioned? How fast can they be made available when they need to be reconstituted?
So the data that's publicly available shows that the French and German products are both reconstituted in less than 5 minutes, a ballpark of 3 to 4 minutes. The Velico product, similarly, about 3.5 minutes. The solvent detergent plasmas may take a bit longer to resuscitate. I haven't seen robust data, but something in a little bit, few more minutes.
So almost immediate availability, 3 to 5 minutes or so for a dried plasma product. Now, in hospital, most centers are utilizing thawed plasma. There's different times they can be thawed in different areas. In the U.S., it's up to 5 days. When a thawed plasma product is used, it's often at day 4 to 5 to minimize waste. So can you compare the hemostatic function when measured in vitro between the day 4 or 5 thawed plasma product compared to a reconstituted dried plasma product?
Yeah, you absolutely can. And there's no doubt that factors 5 and 8 drop off significantly during storage, even out to just 5 days. And if you wanted to use something like a never frozen liquid plasma that can be stored theoretically out to 40 days if collected in CPDA, at least in the United States, the clotting function of that drops off quickly and stays reduced for its entire shelf life due to that loss of factor 5 and factor 8. Does that make it a useless product? No. Clearly, it still has some utility. But compared to a freshly reconstituted dried plasma that in the process of approval has met the FDA standard for what plasma should look like with a full complement of factors, clearly is going to be better.
In addition to the factors that you mentioned, 5 and 8, actually there's been a few manuscripts or publications indicating thrombin generation is dramatically reduced in the thawed plasma product at day 4 or 5 versus a reconstituted dried plasma product, as well as viscoelastic assays too. So yes, we all know in vitro assays may not completely represent in vivo hemostatic function, but it's pretty clear when you compare a reconstituted dried plasma product, which is basically equal to a day 0
exactly
plasma product, and there's no storage lesion that occurs with a dried plasma product compared to the in-hospital standard of care, which is currently thawed plasma, and in the U.S. at 4 to 5 day product. Its hemostatic function is quite poor relative to dried plasma. So the question is, of course, pre-hospital, it's kind of a no-brainer. It would be helpful as an adjunct to whole blood or in isolation if whole blood's not available. But why should we be looking at in-hospital use? My view has always been, if I can get a higher quality product immediately, why would I want to either wait for plasma to be thawed or use a thawed plasma product that has a day 4 or 5 storage lesion associated with it? And I think the question comes down to cost. But can you just talk a little bit about in-hospital use and its potential benefits relative to the cost aspect of the equation?
Well, so it's funny you mention this because in the parts of Germany served by the Red Cross region that manufactures the German product, the Lyoplas, they actually do use the dried plasma in the in hospital settings, and they put it in their sort of Pyxis type system in the ICUs and in the emergency room for all the reasons you just mentioned. I mean, all things being equal, would you rather get the product that has all of the factors and thrombin generation potential, et cetera, or the one that's clearly depleted? I mean, if I were the patient, I wouldn't want the day zero, if you will, product, right? And so German clinicians have adopted it that way. And I think there's potential for that in the US as well. Thawed plasma comes at a cost. If you're a super high volume center, you may not waste very much. If you're a medium volume center, you're going to waste some. There's the quality issues. You still have to have a refrigerator in place. You could be using that refrigeration space for other products like whole blood or even red cells or platelets.
Yeah.
So there are trade-offs that have to be considered in all this.
So you're an advocate for in-hospital use of dried plasma?
Absolutely.
What about future directions? Where is the industry or the industry as a whole going? What additional processing can be incorporated to further improve its efficacy and safety? And then what about combination products moving forward?
Right. So, I think in terms of the plasma by itself, there's a lot of interest in incorporating pathogen reduction technologies to further improve the safety of the product. There's been discussion of engineering plasma or choosing donors who have particular coagulation profiles that might be helpful in the resuscitation of bleeding patients. So those are all things that are being discussed. In the long term, I think that the experience we had in World War II showing that at the end of the day, plasma was great, but it was still a bridge to full resuscitation. You need the oxygen carrying capacity, you need the platelet function. And so in the interest of making a product that was still going to be, still available in the pre-hospital setting, that has all that functionality, that could be shelf stable, the U.S. Department of Defense and other funding agencies have invested significantly in incorporating synthetic or semi-synthetic adjuncts to the plasma to give it those other functionalities. So, for example, artificial synthetic platelets or red cells would be the addition that would kind of, be the whole package in a more shelf stable format. So I think that that's the ultimate future where this is all headed.
And what I think is really exciting about that potential, where you have dried plasma, dried oxygen carriers, and dried platelet type products, you now can reapportion them in a way that is specific to a certain type of coagulopathy. Right? We know the coagulopathy with traumatic brain injury is different than without. We know postpartum hemorrhage, the coagulopathy that occurs there, or the blood failure that occurs there, is different than a liver transplant patient. So what I think is really exciting is that this approach allows us to customize,
right,
or make a precision transfusion medicine application, because whole blood is whole blood. It's how Mother Nature made it in humans. But there's a chance to improve upon that, to directly address the deficits that are occurring in our severely bleeding coagulopathic patients, to give them a little bit more of what they may need according to the etiology of their bleeding. So that's hopefully, it definitely is a direction that the industry is moving towards in the future.
I think so. I think diagnostics will have to catch up, which is a major challenge, but I think that's right. Ultimately, precision medicine is where we'd love to go.
Yeah.
Yeah.
That's a wrap for this episode of the THOR Table of Knowledge podcast. A big thank you to our sponsor, Velico, for supporting the advancement of trauma care. Their frontline ODP spray-dried plasma system is paving the way for blood centers globally to produce potentially life-saving plasma for transfusions. Check out our website, www.thornetwork.com, for more podcasts, and join us next time as we continue to explore the cutting-edge science of trauma, hemostasis, and oxygenation. Until then, stay safe and stay informed.